Good evening, everyone. Am I audible? Yes, can anyone tell me if I'm audible?
Yes, you are audible.
Okay, we will start in a minute. Yeah, so can somebody give me the uh, recap of what we had done in the last class? Is anyone? Okay, let's start first with a few minutes on what we had done so far in the last class. Yeah, so in the last class, we had seen why do we need to have water. Just give me a moment. Okay, so we had seen why do we need water treatment, what pollutants cause uh, hazards, and water uh, unfit for different applications. Right, then we had discussed on different types of pollutants and their classification. Basically, we started off with physical parameters that of water that are uh, that might cause uh, change in the properties of water and that might cause pollution. Then there could be chemical parameters and biological parameters. So far we had seen physical parameters and a few of chemical parameters, and we will go further uh, by continuing this. Also, we had seen various laws and standards which are uh, applied for different pollutants depending on the application and depending on the field, right? Uh, okay, so it could be for drinking water, you will have some different standards. If you want to apply that water for industrial use, then the parameters in the water permissible will be different, industrial purpose. So depending on the application, these standards are also different, right? So till this point we had covered in the last class. Now, if you have any doubt so far, otherwise we will move to the further slides. Yeah. So let me ask a few questions on last class. So what can you name a few physical parameters and chemical parameters in water? Yes, anyone? Um, See, it's better if you reply, okay, right? So that I can get to know. Um, Otherwise, there might be some network issue also, and I will not be able to even know. Yeah, so let me tell you. A few physical parameters that we had seen were temperature, pH, uh, not pH, temperature, color, odor, taste, right? Then we had seen a few chemical parameters also. And now let's start with another parameter, which is alkalinity. Now, alkalinity is nothing but it is a measure of the ability of any solution. Here we are talking about aqueous solution, obviously, but it can have other things. So, not only water, but any solution, water-based solution to neutralize the acid. Now, it could be any acid, HCl, HNO3, H2SO4, but for our safer, for standardization, we can fix the acid which we are using. Now, how, there are multiple ions, H plus, OH minus, then carbonates, bicarbonates, sulfate, sulfide, phosphate, nitrate, etc. Right? So all of these ions, anions, and like these anions, they have tendency to react or to neutralize this H plus, right? Because these are negative, so they can react with H plus. For instance, this OH minus, it is a basic, like it is an anion which has basic tendency, right? It is a well-known thing. Now that it can react with H plus to form H2O, right? Now this carbonate, for example, CO32 minus can react with H plus to give you bicarbonate. Further, this bicarbonate can react with H plus to form H2CO3, right? So all these sort of reaction, what are they doing? They are consuming H plus, and hence you see the acidic nature of any solution, which is because of the presence of H plus, it will reduce, and hence the solution will tend to neutralize. So in that case, we can say that yes. Can you mute yourself? Yeah, so in that case, what happens is since the ions in the solution they are causing reaction and neutralization. So alkalinity it helps in maintaining the pH, right? Because the ions are neutralized, and hence we can say it causes like it acts as buffer sometimes, right? What does buffer mean here? Buffer is any solution like which has the tendency to maintain pH in a particular range, right? So that is what happens here. Now, as I mentioned that these ions, hydroxide ions, sorry, it has to be hydroxyl, right? OH minus, bicarbonates, carbonates, nitrates, etc. They contribute for alkalinity because they have the tendency to neutralize H plus, right? So, and H plus will be subtracted. So, alkalinity is nothing but if it is in mole per liter, then summation of these anions minus the H plus, right? Now, carbonate here is 2 because because of the valency difference, because it can neutralize two hydrogen ions, right? Now, if I have phosphate present, it can neutralize 3H plus, right? In subsequent reactions. So you can have three phosphate concentration, right? So in similar manner, you need to multiply with the valency, right? OH minus, since it is single, it is multiplied by 1, bicarbonate by also 1. Now, always this alkalinity and hardness and everything, they are measured in terms of milligrams per liter of calcium carbonate. Now, the reason for this we had discussed in the last class also because calcium carbonate has molecular weight 100 and we have a basis so that it is always easier to explain and it is always easier to convert. So here in this case, what we are doing, if we want to convert this alkalinity in calcium carbonate, in terms of calcium carbonate, what we do, we take this concentration of any uh, X in which regards we want to measure the alkalinity or the entire alkalinity in milligrams per liter, it has to be, right? But here, if we are talking about X, then it might have different valency. So in that case, we need to have individual ones. Otherwise, if we want to have total alkalinity to be represented, then it has to be in milli equivalent per liter because it normalizes the valency, right? So valency do not come into picture. So here again we are doing the same thing. We are converting this concentration which is in milligrams per liter into equivalent concentration which is milli equivalent per liter by dividing it with the equivalent weight of that particular X, which is nothing but any anion, and multiplied by the uh, 50, which is 50 milligrams of calcium carbonate, which is present in 1 milli equivalent, right? So, how it is obtained? So, 100 is the molecular weight of calcium carbonate, which has valency 2, right? So, to obtain the milli equivalent weight, we have 100 by 2, which is 50. Okay, is it clear so far? Yeah, so this is how we calculate, or this formula you can directly use for measuring the alkalinity. One thing you need to always take care is the unit. You need to have unit consistency every time, right? So either it is milligram per liter or it is uh, milli equivalents per liter. Um, sorry, some I somehow disconnected from the uh, meeting. So it's better in that case if you keep on um, interacting in between, right? So that I get to know if this happens. Okay, let's continue. Is it clear, Sophie? Can you tell me where did I stop or where I got disconnected from the meeting?
From alkalinity, PO4 3 minus neutral, that I can see.
Sorry, acha. Yeah, so I guess from how to convert this alkalinity or the concentration of X in terms of calcium carbonate. I guess that I was discussing, right? Yeah. Um, is that you were speaking? Uh, yeah, milligram of per liter of X. Yeah. So, yeah, how do we convert the concentration of any uh, X here represents any of these anions in terms of calcium carbonate. So first step is to uh, convert milligrams per liter of X to milliequivalents per liter. And how do we do that? You divide.
Excuse me, ma'am. Screen is not visible, ma'am. Please.
Oh, sorry. Give me a moment. Yeah. Is it visible now? Yeah. So in that case, what we do? We divide this concentration by the equivalent weight. So let's take some example. Here, let's take example of calcium, this carbonate. So please somebody help me to calculate what is the molecular weight of this carbonate ion. Molecular weight. See, I might not solve.
62, ma'am. 62, ma'am.
Yeah, can you expand? How have you done so that everyone knows?
Carbon 12, carbon molecular weight is 12.
Yeah,
Oxygen molecular weight is 16. 3 into 16 plus
yeah.
2.
Plus two
Plus
for
2.
what?
Charge mele 2. Mele
No, no.
charge.
You,
Charge.
while calculating molecular weight, you do not include charge, right? So it's
Okay, okay,
just
okay.
carbon plus oxygen. So it's 12 for
Oxygen
carbon
48 plus yes.
plus 3 into 16, 48. So do
8
that,
plus 12,
yeah,
60.
right? So 60 is the
Yes.
molecular weight of carbonate. Now this is the molecular weight. Now how do you calculate equivalent weight? So equivalent
Divided by
weight.
valency.
Wait, yeah, you are correct. So it is 60 by 2. Here the valency
2.
is 2, right? So its
Yes.
equivalent weight is 30,
30.
right? Now, So its unit will be milligram per milli equivalent or gram per equivalent, right? So now to find out the concentration in equivalent, milli equivalent per liter, what you do is, let's say the concentration is X, so you divide it by 30, right? Do you get this point? Till this point, is it clear?
Yes, ma'am. Ma'am, I
Yeah.
have one doubt. Alkalinity means only the base ions only we can consider, ma'am.
Yeah, base
CO3
ions
minus
only.
hydroxide,
Yeah,
bicarbonate, nitrate,
because
and then ammonia.
yeah. So all the basic ions you can consider because it has the tendency to neutralize the hydrogen, right? So say for example, it's CO3 minus. So since it is anion, basic anion, it can react with H plus to give H2CO3, right?
H2CO3, carbonic acid.
Right?
Yes, okay, ma'am.
So this is a
Yes, ma'am.
neutralization reaction which, because of the presence of this bicarbonate, it can undergo. So that is how neutralization occurs, and we can say it contributes towards alkalinity. So
Okay,
similar reactions you can write for other anions as well, or other basic ions. Okay?
okay, okay.
Yeah. So yeah, now if you want to further convert this into in terms of calcium carbonate, you just need to multiply the equivalent weight of calcium carbonate. Now let's calculate the equivalent weight of calcium carbonate. Can you help me with that? It's easy. First, calculate the molecular weight of calcium carbonate.
Calcium 40, calcium 40, carbon 12, oxygen 48. 50.
Yeah, so it will be 100, right? So in that
Yes, ma'am.
case, here the valency is that you need to
2,
consider is valency
2.
2. So
Uh,
equivalent weight will be,
100 divided by 2 equal to 50.
yeah, right. So here to convert it into milligrams per equivalent or milli equivalent per liter of calcium carbonate multiply by 50, which is a common factor for every case. I guess it is understood, right? So this formula will help you this too in calculating the alkalinity, right? And one more hint I would like to give is whenever you are adding any concentration, right? So you need to check whether the concentrations are in same unit. For example, if this carbonate is in milligram per liter and this bicarbonate is in milli equivalent per liter, you cannot add, right? Or let's say this is in milligram per liter, this is in molarity, you cannot add. Okay, so always ensure that the units are same while you are adding or subtracting any concentration. Is it clear? Yeah, so let's move forward. How do we determine experimentally the alkalinity? So in that case, what we do for any solution which has pH greater than 5, you titrate it with acid such that you uh, reach pH 4.5. So from that, how much amount of acid you require to neutralize, that is corresponding to that alkalinity. And similarly, if it's greater than 8.3, then you lower it till 8.3. Like these two are just standards or 2 points so that you can have some measurement or you can have some thumb rule. Okay, so these two are for some thumb rule. Now let's move forward for the next type of parameter, which is dissolved gases. So dissolved gases can include oxygen, carbon dioxide, hydrogen sulfide, ammonia, etc. Right? So where does this gases come in water? So oxygen, if we talk about, then oxygen is present in atmosphere, which is equivalent to 21%, right? So you would be knowing that there is some solubility of everything. Like for example, even gases has certain solubility in water. Salts also are soluble in water. So these gases, say for example, this is the water surface and this is atmosphere. Here it is oxygen 21%, there will be even carbon dioxide, and this is water. Now, obviously, water has some solubility, and depending on the concentration, like here, obviously, it is more concentrated, here, it will be less concentrated. So, mass transfer happens. Okay, so the transfer of oxygen happens from atmosphere to the surface of water. So, water on the surface, it absorbs oxygen or carbon dioxide, and later since inside the water also, like this is water bulk and this is surface of water. So from surface once it comes on the surface, it travels to the ox- uh, bulk of water, right? So what happens is, let's say from atmosphere it has come to this surface. Now again there is concentration gradient, like this has, let's say, uh, x milligrams per liter. Always the concentration in water is expressed as milligrams per liter. So x milligrams per liter of oxygen on the surface, while on the bulk it will be some, let's say, initially it is zero, initially. So what happens? It will start migrating, like it will have mass transfer from the surface to the bulk until a uniform concentration is maintained. So after certain point of time, here the concentration will be, let's say, Y. Again, here also will be Y. Now, this oxygen will be further consumed by all the aquatic and bodies. For example, fish, they consume this oxygen, and the oxygen deficiency which might be created when more oxygen is consumed, then it again comes from the atmosphere. Another source of oxygen can be the plants in the water bodies. So during respiration, they release oxygen and contribute towards the dissolved oxygen in the water bodies. Now, what is the standard for dissolved oxygen in water bodies? Can you tell me which is required for all the living aquatic living organisms for their healthy life? Do you know what is that concentration? Yes, anyone, do you know? For example, uh, um for us, because we are inhaling and exhaling from atmosphere, 21% of oxygen is required. So what is that concentration in water bodies? That is required for aquatic organisms because they inhale and exhale from the water itself. Okay, I will tell you. It should be around 8 milligrams per liter for healthy uh, aquatic animals, right? Now, next is carbon dioxide, right? So, in carbon dioxide, it again comes from some um atmosphere, and it is required for the algae and the plants for their respiration. Now, Again, this carbon dioxide, what it does? Now, this calcium ions and magnesium ions, they are responsible for hardness, right, in water. So this, they can interact with the carbon dioxide which is present in water to cause the hardness and also the precipitation of calcium carbonate and magnesium carbonate. So this also can be formed. And there
Ma'am,
could be,
oxygen is also helpful for the decomposition of organic matter into inorganic matter. Yes, ma'am.
yeah. So,
Or
yeah, you are very right. So, if there is any organic waste, even if natural waste, for example, excreta of any living organism, so that is also organic, right? So, it is CXHYOZ. So, for that also, oxygen is consumed by all the microorganisms which are present in the water body, and they are converted again to carbon dioxide and water. Now, again, this carbon dioxide is, consumed by the algae and plants for their growth. So that there is a circular, a cyclic process.
cyclic
Let's,
process.
yeah, so plants.
So, ma'am, this process is called biological oxidation, ma'am.
Yeah, so naturally, whatever is occurring, it is a biological process because the naturally
Okay.
occurring microorganisms, okay, they, okay, so these are microbes. They consume oxygen which is coming from the plant while they are exhaling. They convert organics to CO2, which is again
Yeah,
consumed by plants.
mitochondria.
So, right, this is cyclic process. Now there are like for this microbes they are used to it, right? Because the naturally coming excreta and the
Yes.
plant waste, for example, plant dead bodies, animal dead bodies, they are also organics. So these everything is degraded by these microorganisms and a circular cyclic process comes into picture. Now, in case if we dump any wastewater which also contains organic, then there are chances that this oxygen and carbon dioxide levels are changed, right? And it disrupts the natural process and the natural cycle which is going on. In that case, that it creates toxicity. Again, if we are disposing the organics which are non-biodegradable, for example, these microbes cannot act upon it to degrade it. In that case, they can further cause bioaccumulation. Do you know what is bioaccumulation? Yeah, so in next slides or only we will be talking more on what are biodegradable and non-biodegradable organics. But just let me tell you in brief that there are two type of organics. One is biodegradable. Uh, so biodegradable are the ones which microbes can degrade. For
Ma'am, can
example,
you explain BOD, COD concept, ma'am?
yeah, so next slide is only BOD, COD. So should I touch upon it
Okay.
later? Yeah. So for now, you just concentrate on where carbon dioxide is consumed and how the cyclic process goes on in the water body. And next is chlorine. So chlorine is usually in the free form, which is Cl2, hypochlorite ion, HClO minus, HClO4. Such ions, they correspond to free chlorine. And this HClO4 also, it is uh, how does this chlorine disinfect water? Because this is widely used for the disinfection of water. Why they are used? Because they form free radicals, and this free radical they are highly energetic, right? Because they are very short-lived and they have only one lone pair of electron. That is why they are highly energetic, and they want to have more and more reactions they want to encounter, right? So in that case, this energetic ions they interact with whatever is present. So in that case, these energetic chlorine ions, they are found to cause disinfection by killing the microorganisms, right? Because microorganisms can cause, you know, disease to any living organism. Now, in the case where you want to have the detection of chlorine, because whatever you are giving chlorine dosage, so some amount will be left for which is unused, right? So in that case, there is a starch iodide test where you add potassium iodide and starch. Now here, this iodide is firstly I minus, which is converted to I2, right? So it is a conversion. Now this I2 in the presence of starch, like this will be in the absence of starch, and in the presence of starch, it gives dark blue to black color, right? Now this is the color in the because of the formation of iodide in the solution. Now the amount of iodide formed, it depends on the Cl, right? Because Cl is responsible for conversion of iodide to iodine gas. Now again, to find out how much amount of iodine is present, we need to have color change, and for that we add thiosulfate. Okay, so here we add thiosulfate, which converts this iodine back to iodide, and again the neutralization occurs. So again the color transformation occurs, and you see how much thiosulfate is getting consumed to neutralize this iodide, and by that you can calculate what is the um, you know the amount of chlorine present. Now, if we talk about another parameters in water or another pollutants in water, it could be iron, lead, arsenic, cadmium. Now, these are pollutants. Iron is even required by our body, but if it exceeds some amount, it can even act as pollutant and harm our body. While these are not at all, these are toxic even at very low concentrations. Low concentration or even at trace concentrations, right? So it is advisable that it should not be present. Now, how do we measure these metals? You can use atomic spectroscopic techniques. For example, atomic absorption spectroscopy, uh, inductively coupled plasma atomic emission spectroscopy, mass spectroscopy, etc. Right? So it will give you the concentration in ppm of each metal, which is nothing but milligrams of, for example, if we are talking about iron, then milligrams of Fe present in 1 liter of water. Okay, so this is how you interpret ppm, or another representation is parts of Fe per unit million part of uh, water here, right? Okay, now let's move forward to BOD. Now, BOD and COD, they like BOD here is the biological. So from now, whatever the parameters we are talking are the, uh, you know, this, it is BOD. How, like, why is BOD counted, and why do we need to measure it? Uh, what do you know about BOD? Can you tell me? You were asking about it.
Biochemical oxygen demand, ma'am.
Yeah, so it is a biochemical or a uh, biological oxygen demand. Okay, so it's
Decompose that in organic to inorganic matter.
yeah,
Oxygen required for how much amount of oxygen required for convert organic matter into the inorganic matter.
yeah. You cannot. Yeah, you are correct. So whatever here we are considering BOD means it is the measurement of the oxygen. Okay, so we are not directly measuring the organics, but indirectly it is represented in terms of how much oxygen it is required to completely degrade the organics into CO2 and H2O, right? Now here the organics they are degraded by microorganisms, right? So microorganisms they are added and they act upon the organics and they convert them into CO2 and H2O. Now here since microorganisms are coming into picture, so microorganisms they act upon organism and that is why the whatever the organics which can be degraded by microorganisms they are termed as biodegradable, right? So biodegradable means it can be naturally degraded. We do not need any specific treatment or any special chemicals, special oxidizing agent to convert, right? If we add microorganisms, they can degrade them. So what happens is we can reduce the toxicity of these organics, right? So what happens is even we can have nitrogen as well, right? So nitrogen can also be converted into NO3 minus, which is the least or the highest oxidation product that can be formed, right? So there can be two stages of BOD measurement, which is carbonaceous and nitrogenous. Here, so first what we observe is firstly all the carbon is oxidized and later nitrogen is oxidized. So in the BOD measurement, we only consider the requirement till carbon for what is the requirement for carbon, right? So how do we measure BOD? So let's say you have a water sample in which you want to measure BOD. Now in case of BOD measurement, wastewater samples they have very high BOD and BOD is nothing but the dissolved oxygen requirement, right? So it is measured in terms of milligrams per liter of O2 required, right? So usually BOD is since they are very high concentrations of organic and wastewater, hundreds or thousands of milligrams of O2 is required per liter of water to completely oxidize, right? So here what we do, it's very difficult to degrade such high concentration or in lab if we want to monitor, it's very high concentration of VOD, right? So in that case, what we do, we dilute this water sample. And why is dilution required? Because not only this BOD concentration is high, the naturally occurring water dissolved oxygen concentration is 8 milligrams per 8 to 9 milligrams per liter, right? So in that case, usually we maintain BOD which is always lesser than this concentration. Like oxygen required is always lesser than this. So this dilution factor is P here, right? So how much? So what we do, let's say we have a water sample in which the dissolved oxygen, so earlier the dissolved oxygen concentration was DOI. And now after the degradation of all the organics, The final dissolved oxygen remaining is the DOF. Now the water sample which we had used was diluted P times, right? So for P times diluted BOD, okay, for example, let's say the concentration of organics or the BOD was actually 100, right? But we have diluted it 100 times. Okay, so 100 times dilution means what does it mean? Can you tell me what is the significance of 100 times dilution here? What does it mean? Do you know or you don't know? Okay, let me tell you. For example, you have any pollutant. Let's say its concentration is X milligrams per liter. Now, how do you expand this X milligram per liter? It is X milligrams of pollutant per 1 liter of water, right? Now, if you want to dilute it, so what you are doing, you are adding, let's say, 1 liter of pure water. So now this X milligrams of pollutant, it is present in how much amount of water? It is present in X milligram per 1 liter was already there, right? And you are adding 1 more liter, right? So 1 plus 1 liter of water. So what happens is now the X milligrams is present in 2 liters of water. So the final concentration is what? Earlier the concentration of was X milligrams per liter. Now the concentration will be X by 2 milligrams per liter. Now this is known as dilution, right? But in real scenarios, what happens is the instruments which we use to measure any pollutant concentration, those instruments might not be accurate enough to measure, for example, 1000 milligram per liter or 2000 milligrams per liter. These are nothing but instrument limitations, right? So in that case, what we do, we need to take this 100 or 1000 to such a limit that we are in the measurable range. For example, if I am talking about, let's say, lead, then the measurement is usually performed from zero point zero 0.01 ppm to 100 ppm. Let's say this is the instrument limitation. Now, what if we have 1000 milligram per liter as the or 1000 ppm as the concentration? So here ppm and milligram per liter are same, right? I explained it earlier. Now 1000, it is since out of the range of instrument, I dilute it first. I dilute it such that it's, let's say, 10 ppm. So the diluted sample I am getting as 10 ppm. So how much times I have diluted? It is 1000 by 10, which is 100 times dilution. So after 100 times, I am getting 10 ppm. So the actual concentration is 10 ppm into the dilution factor, which is, let's say, we are defining it here, P. Are you getting my point? What I am saying? Anyone, can you confirm? What is dilution factor and how do we multiply it? Why do we need to multiply it? Yes, anyone? Rajeshwari.
Yes,
So, are you getting my point, or are you understanding the concept of dilution and how dilution factor is multiplied?
Yes, ma'am. Initially taken that it can be diluted, that is the X divided by 2 milligrams of the mole. Yes, ma'am,
Sorry,
I understand.
you. Okay, okay. Yeah, so that is how this comes into picture because while measurement we dilute the water samples, let's say by P times. So for P times diluted, while the actual concentration is this much, P times dilution, the change in dissolved oxygen is DOI minus DOF. So the actual will be you divide by P, like this P goes in the denominator, right? Now there are different terminologies. BOD can be measured. How do we measure BOD? In an incubator, right? So we add the microorganism seeds, right? So here if you take microorganism seeds in the water sample, so even some dissolved oxygen can be consumed by the water sample itself. So that is also subtracted, which is the um, oxygen consumption because of the microorganism in the absence of any organics, right? So that is subtracted here, that correction is taken into account. Now for that what you do, you after adding the microorganism you add now these microorganisms they can uh, have the maximum activity at a particular temperature and temperature condition, which is 37 degrees Celsius. Okay, even if you know our body is also in the range of that temperature. So what you do, if you want to do 37 degrees Celsius, maintain that temperature, you put it in an incubator which is maintained at that temperature. So that bacteria and microorganism they have that environment to grow, right? And show the degradation. So in that case, uh, the time required for degradation, you maintain a fixed time after which you will measure the oxygen concentration. So it can be 3 days or it can be 5 days also at 37 like at 37 you maintain 3 degrees 3 days. For 5 days you need to maintain if it is 20 degrees Celsius, right? So depending on the temperature you can maintain BOD 5 is 5 days BOD measured after 5 days. If you are measuring after 3 days, then it is BOD 3. Now, how it is derived, the BOD formula with respect to time, and what is the rate of it? So let's say the here the L is the rate at which the degradation of organics happen, and let's say the organic concentration is determined by L. So at any time, let's say L is the concentration. So its change in because it is reducing, so dL by dt equals to minus because of the reduction. K is the rate at which rate constant, okay, specific rate constant into the concentration. So it is a first order reaction because LT is directly dL by dt is directly proportional to LT, like sorry, L to the power 1. Okay, so first order reaction. Now, if you want to solve this, you need to integrate it, right? So 1 by LT dLT equals to minus KT, KDT, right? So on integration, if you want to integrate and put the limit, just give me a moment. So from LT initial to final L naught, sorry, initial L naught to LT. L naught is the initial and final at the end of time at any time. Let's say it's LT into, sorry, it will be 0 to T. So if you integrate, you will get this formula, which is LT equals to L naught e to the power minus KT. Now, if you take the summation of all this, then L naught is equals to LT plus BOD at any time T, right? So now you put up what is LT and finally you will get BOD in terms of this organics, which is nothing but L naught 1 minus e to the power minus KT, right? Is it clear so far? Okay, so now let's move to next slide, which is COD measurement. Now, as I mentioned that microorganisms were used earlier to degrade the organics. Now, microorganisms cannot degrade all the organics, right? Say, for example, you have pesticides, you have pharmaceutical products, then you have other recalcitrants. All these are microorganisms cannot degrade, right? For example, nowadays, even micro and nanoplastics are also present, which are nothing but solids, but these all are not degradable by microorganisms, and hence they are known as non-biodegradable, right? Which are not degradable by microorganisms. So in that case, you use a chemical which is, strongly, which has strong oxidizing potential, oxidation potential, and hence it can readily oxidize all the types of organics which are present, right? So here the BOD, which used to take 5 days or more for measurement, here you just need 1.5 to 2 hours because you are using strong chemical for the oxidation. Right? So here both biodegradable and non-biodegradable organics, they get converted into CO2 by using potassium dichromate, which itself gets reduced from Cr plus 6 to Cr3, right? And because of this strong oxidation condition, all are getting converted. Now COD is always greater than BOD. Why? Because here COD, both biodegradable and non-biodegradable are converted, while under BOD only biodegradable ones are considered, right? Now, if you want to measure what is the amount of oxygen requirement or the what is the amount of organics, so then in that case you first know what is the K2Cr2O7 amount which you had added, right? Now, depending on the organic concentration, this K2Cr2O7 will be converted to Cr3 plus. So to measure the leftover Cr2O4 2 minus, like which is Cr3 plus, you add Fe2 plus ferrous sulfate, right? So on adding ferrous sulfate, this ferrous sulfate will be converted to ferric, which is 3 plus, right? By leftover Cr, Cr plus 6. Okay, so leftover Cr plus 6 will convert this ferrous to ferric and this Cr plus 6 to Cr plus 3, right? So this redox reaction will occur, and based on the titration amount, because you know what you had earlier added, and now you can know how much it is consumed by the ferrous, right? So the amount consumed by organics is equals to initial minus the amount consumed by Fe 2 plus, right? So by this calculation, you can you are not asked to go in deep of this calculation. I'm just giving the mechanism how it is done. Okay, so the leftover will be the amount consumed by organics. Now another is parameter is total organic carbon, which is nothing but total carbon which could be contributed by both organic and inorganic, minus the inorganic carbon which could be because of the carbonates and bicarbonates, right? So if you subtract that, you can get the organic carbon. Now the theoretical oxygen demand is nothing but the oxidation is the amount of oxygen which is required to oxidize to the final oxidation products. And here the final oxidation products will be carbon dioxide for carbon, nitrate for nitrogen, and sulfate for sulfur, right? So if any organic compound is present, for example, CA, C2, H4, then you convert it into CO2 plus H2O, right? Now let's say if you have any compound which has C2H5 in 2. Now this nitrogen is to be converted into NO3 minus. Now if it would be nitrate already, then further oxidation is not required, right? So this is how you need to oxidize the product for theoretical oxygen demand. So this is nothing but the stoichiometric or the theoretical demand for oxygen for the uh, oxidation, right? So whatever you supply in actual process will always be much greater than the theoretical demand. Now let's move forward with another parameter, which is uh, microorganisms. Now in that case, there could be number of waterborne diseases and water related disease, which could be because of for example, waterborne disease, which can occur because of the presence and infections spreaded by contaminated drinking water. Then it could be because of the diseases which lacks sanitation and hygiene, right? Because water microorganisms might be present, but if you are not sterilizing the water and sanitizing or some unhygienic activities are performed, then also some diseases might be uh, occurring. And then you can have diseases because of the organisms already present in water, or some cholera or cholera sort of disease which can be spread where the microorganisms they propagate and they travel through water, right? So such sort of disease are always uh, occurring. So in that case, if you want to measure the microorganisms in water, you measure it in terms of the organisms which are easy to measure, and they represent a sort of all types of organisms. So in that case, you do the measurement of fecal indicator bacteria, and which is nothing but uh, used to check the presence of pathogens in water. So it is the indicator of the microorganisms which are harmful to us, that they are present in water. So in that case, uh, what does this fecal bacteria mean? Fecal bacteria is nothing but it is found in the intestines of warm-blooded animals, for example, human beings, some aquatic mammals, right? So birds, they are present in them, in the intestines of those organisms. And E. coli is one such, bacteria which is used to measure the microorganisms present in water. Now, how do you measure this E. coli? Okay, so there are different methods of measuring. What one is plate count, another is membrane filtration, and last is most probable number. So here you see the this is a petri dish, and here you see this white dots. These are nothing but E. coli bacteria present, right? So for that, what you do? Firstly, let's say you have a water sample. Now that water sample, as I mentioned, you do the dilution because there could be lacks of bacteria, and obviously we will not be able to count it. So in that case, what you do, you dilute it such that we can have it is readable, it is measurable, or it is countable by us because you need to count on your own. So what you do, bacteria obviously requires some nutrients for their growth. So you pour a petri plate with agar and broth, which contains all the nutrients for the bacteria growth. Now you take water sample, usually in microliters, and you drop it, and now you scrap it. So this individual dot is nothing but one colony forming unit of bacteria, which is nothing but almost one bacteria or one microorganism, right? One E. coli cell, you can say, which has grown. So now you count all these cells, and whatever you get is nothing but number of colony forming units, CFU, per ml is the concentration, right? So this many bacteria or this many microbes, you can say they are present in this water, right? So ideally, if you want to drink water, it should not have any microorganism, or E. coli should not be present. Okay, so that is the representative of fecal bacteria or fecal microorganisms, and since they are harmful, it should be zero. Now another is my membrane filtration, where you take a membrane, you sterilize it because other microorganisms can also be present. So you sterilize it so that other microorganisms do not affect the growth of of the water sample, and you dip it. Now you provide all the nutrients and you dip it in the water sample. After that, you allow that membrane uh, for the growth of bacteria by providing it in the agar and broth, and now you count the colonies. So that is how you do membrane filtration method. And last is most probable number method, where you dilute your water samples such that you have one solution which is known as broth. Now you there are test tubes in which broth is placed, and the water sample is also allowed to interact. Now, if your bacteria is present, then it will form bubbles, while if the bacteria is not present, the bubbles will not be formed. So, here bubble formation is the representative that um, you know bacteria or the microbes are present, and there are standards available that for this much amount of bubble formation, this is the concentration of uh, microbes which is probably present in that sample. But mostly you should do plate count and membrane filtration to get the clear idea of how much amount of microbes they are present. Right? Is it clear so far? Yes. Okay, I assume it's clear. So this charts, they are nothing but the summary of all the parameters that we have talked, and how do we measure, or if any specific instrument is available for that measurement. So that summary is given to you, which was already discussed by professor, and I will not go into much deep of it. Okay. So now once we have discussed all these parameters, we have known that what could be the toxic impacts of these pollutants because of their presence in water if we consume that, and what other drawbacks or probable disadvantages you see if they are present in the environment, and what harmful impacts on ecosystem can also be observed, because not only health hazards of human is the prime importance, entire ecosystem needs to be considered, right? So obviously we need to treat that water, and we need to either remove the pollutant so that at the end, if we are consuming that water, it is not harmful to us, right? So there could be different treatment strategies, and depending on the end use. For example, if you are using uh, a water filtration unit or a reverse osmosis, which is RO unit, mostly found in every household nowadays. So if we are using that system, it is point of use system. Now, if you are implementing a filtration unit in a village where everyone uses it for the drinking purpose, right? So everyone collects water from that point. So it is a community treatment. Okay. So all the people, they depend on the that filtration unit which is available for the entire population which is staying nearby, right? So that is community treatment. Now, if we talk about, for example, even if we talk about households, then there are multiple points where we generate water. For example, if we are washing clothes, if we are washing utensils, if we are bathing, from toilets, um, from kitchen, from everywhere, water is generated which contains some amount of pollutants, right? Obviously, if water is used for washing utensils, that water coming out from there, we will not use it for drinking purpose, right? So that needs to be treated, right? So where does that water go? So that water is nothing but known as municipal wastewater, and obviously, or sewage, right? So if you want to treat that water, in every district there are treatment units where you treat this water, right? So this type of water usually contains salts, um, surfactants, and biodegradable organics, pathogens, right? So these are the, and some, yeah, suspended solids also, and some colloids, right? So all these are the most probable pollutants which are present in municipal wastewater. So only one wastewater treatment unit will be provided for that, and every district has that. So it is nothing but known as sewage treatment plant, STP, or common effluent treatment plant, CETP. Mostly for municipal wastewater, it goes to STP only, right? I will tell you what is a CETP. This was about if the wastewater is generated from household usage from commercial buildings, etc. But what if the wastewater is generated from industries, right? So this specific to industry, the pollutants also concentration also varies, and also the type of pollutant present, they both vary, right? So there could be some industry specific wastewater treatment plant. For example, if you talk about big or large scale industries like Reliance, they have their own wastewater treatment plant, which is nothing but known as effluent treatment plant. So wastewater coming out from industries is nothing but known as effluent. And the treatment plant for that is nothing but effluent treatment plant. Again, you can have sewage treatment plant in case uh, you have, for example, township, right? For example, Reliance will have its own township and no water will come out of that township, right? No discharge will be allowed from it. So they have their own treatment plant. So sewage treatment plant for that particular township can also be there. Now, this was about large scale industries. What if you have small factories, small industries, or small scale industries? Now, small scale industries, they are not located isolated, but there is a cluster of small scale industries. And you would be knowing that these industries, they are not near to residential areas, but they are in a specific region, which is obviously industrial zone. And in that zone, there are many small industries, for example, textile industries, paint industries, very small units, they are there. And each of this unit, they generate wastewater. Now, because these are nothing but small scale industries, they might not have that much land or they might not have that much money so that they can set up their own wastewater treatment unit, right? So in that case, a common effluent treatment plant is set up in that cluster of small scale industries, which treats the water coming from all the small scale industries in that region, right? So wastewater from all those units, they go into the CETP and where it is treated. Again, CETP only allows the wastewater. For example, there are five industry: one, two, three, four, and five. Now, all these industries, this is, say, for example, textile, so it will have many dyes in it, right? Let's say then you have paint industry, then you have pharmaceutical industry, right? So all these are sort of small scale industries. They are example of it. And there could be many more types of industries. For example, pulp and paper, right? Pulp and paper mill. So all these have different characteristics of wastewater, and since they are small scale, they might not have that facility to treat it completely. So since they have different characteristics, the CETP have will have some standards. Okay, so this industry will need to primarily preliminary treat it such that any specific pollutant, if it is present, they need to take care of it, or they also have guidelines such that, for example, only 1 meter cube per hour is allowed. For example, this is the uh, uh, amount of water which is the maximum amount which this industry can uh, dispose, right, and send it to CETP where it is treated. Also, they need to pay some amount so for the treatment to CETP, right? So that is how CETPs work. Now, another concept nowadays which is considered is ZLD, which is zero liquid discharge. So this concept is considered in almost all large scale industries, all the CETPs, where what you do, you whatever the wastewater is there, so it will have, so you convert that wastewater into pure water, which can be recycled and repurposed into the industry itself, or you can use it either for sanitation and so on. So basically you can recycle and reuse, or and you will form a solid which is generated from that wastewater that needs to be landfilled. So there will be two products, or there will be two products from wastewater. One is liquid phase, which is pure water, and another is solid form, which could be toxic, which could be biodegradable, anything. Like it can be combination of all, right? So solid, these two are formed, and there is no liquid which is coming out of that industry or from that zone. So that is why it is known as zero liquid discharge, which means no liquid should come out of that industry or from that zone, and whatever the liquid water is there, you recycle and repurpose that water, right? So that is nothing but zero liquid discharge. Now next is for treatment of such wastewater, there are basically the treatment steps. They are divided into primary, secondary, and tertiary treatment. So first, this primary treatment takes care of uh, all sort of removal of suspended solids and bulk material by sedimentation and equalization. Okay, so for example, if you are talking about CETP, then wastewater comes from different industries which will have different parameters, right? So what you do, you mix it, which is nothing but equalization. So all the parameters are now uniform, which can be sent further, right? So all such process or pre-processing, it comes into primary and preliminary treatment. Now, secondary treatment is nothing but you remove all sorts of biological parameters, organics, by using or by providing biological degradation. Okay, so secondary treatment is nothing but biodegradation by using microorganisms, which can include activated sludge process, various lagoons, trickling bed filters, uh, biogas plants, right? So all sorts of secondary treatment can be provided. And lastly, whatever is not treated by any primary or secondary treatment, then in that case, we provide tertiary treatment, which is a specific treatment procedure. It can include adsorption, membrane filtration. It can also include disinfection and advanced oxidation process for all the non-biodegradables and so on. So all these processes one by one from next class we will discuss, right? So that is why I didn't go into deeper right now. So right now you just remember there are three types of treatment steps. One is primary, secondary, and tertiary. And all these steps are followed in this manner only. Few process can be added, few can be eliminated depending on the wastewater characteristics. Is it clear so far? Yeah, so now we will move forward with the sample assignment. Okay. So what will be the pH of water sample when it has no alkalinity because of natural ions? So that is pH 4.5. If in any water sample having pH greater than 8.3, addition of phenolphthalein indicator will change the color to pink. Which ion is responsible for the color change? Now, addition of phenolphthalein indicator. Okay, so in the basic pH, it gives pink color, which is because of the OH minus ions, right? So correct answer here is OH minus ions. Now, if this pH is like if this sample is neutralized, then phenolphthalein indicator will give you red color in the acidic medium. So if it is acidic, you will have red color because of H plus ions, right? Yes, are you following me? Okay, so these assignments are very important for all of you because similar questions might be expected from you all in the assignment which is given to you and also in the exam at the end of the semester and at the end of the course, right? So, yeah. Next question is addition of which chemical to the water sample will result into disappearance of pink color indicating neutralization of all the OH minus ions. So disappearance of pink color. So pink color is because of the OH minus ions and that is because now if this OH minus ions are only not present, then the pink color will disappear, right? So for that, if you neutralize it, then it will disappear. Now, neutralization can be because of H2SO4, right? So that is the correct answer. Next is in carbonaceous stage of the BOD test, organic carbon converts into which species? So BOD here the organics are converted to CO2 plus H2O, and if you look into the options, any of this is not having CO2, so none of this is the correct answer. Next question is which pollutant in the drinking water is responsible for few cavities in the teeth? So it is nothing but fluoride. Okay. So fluoride is
Fluoride.
yeah. So fluoride is a nutrient which is allowable till concentration of 1.5 milligrams per liter, right? Above that, it can cause dental fluorosis, right? So it can produce fluorapatites and it can make the teeth even brittle and it can even cause your bones to be brittle. So it has, if above 1.5 ppm, it can cause uh, cavities and it can cause your harmful impacts to your bone and your teeth. Next question is if glucose is completely oxidized to CO2 in aqueous solution, its theoretical oxygen demand is 100 milligrams per liter. What is its COD? So here you see glucose is C6H12O6, right? It's converted into CO2. Now here you see it does not have anything like nitrogen and or sulfur which is considered in theoretical oxygen demand. So whatever the oxidation occurs is because of the C6 only and the oxygen consumed will also be because of C6 only. So whatever the theoretical oxygen demand here you are having, it is just because of carbon and hence it is same as COD because COD will also be the amount of oxygen because of the carbon requirement, because of the carbon. So 100 is the correct answer. So this is only you need to apply your mind. There is no calculation required, right? Because you see the data says if you look into the glucose formula, it does not have any nitrogen and sulfur and that is why the Theoretical oxygen demand and COD will be same. Now again some twist can be, what is the BOD of this sample? So again COD will also be same as BOD. Why? Because this glucose is biodegradable, and that is why COD and BOD will also be same. So here THOD, theoretical oxygen demand, is equals to COD is equals to BOD. Is it clear?
Yes, ma'am.
Okay. Next question is, dash is the measure of the ability of a solution to neutralize acids to the equivalence point of carbonate and bicarbonate. So that is nothing but alkalinity. Neutralization of acids is nothing but neutralize H plus, right? So answer is alkalinity. Next question is, which salts are responsible for hardness in water when they react with CO2? So which ions are responsible for hardness? This is very basic question.
Calcium and magnesium.
Sorry?
Calcium and magnesium.
Yeah, calcium and magnesium, they are responsible for uh, hardness. Correct. Next is, which statement is incorrect? So incorrect, you need to find out. So let's look into the options. First option is, dissolved free chlorine is never found in natural waters. So that is correct. The salt-free chlorine is present in treated water resulting from disinfection with chlorine. That is also correct, right? Because there could be some residual chlorine because some might not be used. Residual chlorine should remain between 0.5 to 0.2 gram per liter in water so that it remains safe against pathogenic bacteria. That is wrong statement because it has to be between 0.5 to 0.2 milligrams per liter, not grams per liter. So this unit is wrong, right? And last option is, so this is incorrect. Last option is, residual chlorine is determined by starch iodide test. So this is correct. So the incorrect one is option C, right? Last question, which parameter gives the oxygen required for the complete oxidation of both biodegradable and non-biodegradable matter? So here the question you need to look into, sorry, the key term in the question is both biodegradable and non-biodegradable you need to consider. So the oxygen demand
COD.
for this is COD. Correct. So the COD is
COD.
correct answer. Right, so that's it from my side today. If you have any doubts so far, you can let me know, or we can end this class. We will meet in the next class on next Tuesday.
Ma'am, please post the detailed notes, ma'am.
Sorry?
Ma'am, please post this
Yeah,
chapter
yeah,
later, the
yes.
detailed notes.
Detailed notes.
Yeah.
So this
Okay.
slide is fine to you?
Okay, thank you.
These slides?
So what you are thought that notes you are given means that's
Yeah, so
okay.
I will share all the slides and again the video also. Uh, the recording of this video will also be on the YouTube. So that link will be provided. You can look into that also.
Okay, ma'am.
Okay.
Okay.
Yeah, thank you for joining.
Okay.
Thank you everyone. We will meet in the next class on
Thank you,
next
ma'am.
Tuesday. Thank you
Thank you.
everyone.